quacking frog actually quacking. So I've got 1 second slices of audio. I'm going to call that quack and then upload it. Okay, and then I've also got some background recordings of typical sort of background noise when the frog isn't there calling. We can upload those. So this is a typical task. I've got a recording of the sound that I'm interested in. I've got a recording of typical sort of background sounds. And what I really want is some sort of classifier that will say, okay, well, is this what I'm listening to now? Is this one of these interesting sounds, or is it a background? And then I can record the interesting sound and ignore the background sound. So Edge Impulse makes it very easy to do that. So I can come along here. I'm going to add a processing block, which is basically a sonogram. I can add a machine learning block. So I'm going to do some classification. I can save this. And then I come in here and I set the parameters of my classification. And so one of the things we're interested in with devices is typically sort of making a model that's fast enough to run on the device and also doesn't take very much RAM. So there's a little bit of sort of tuning that I can do here to sort of change some of the parameters of my model and that of the pre-processing. And that's basically going to control how long this processing takes, because I need it to run in real time, and then also sort of how much RAM it's going to take. So this is basically a spectrogram with a MEL frequency sort of imposed on it to sort of reduce the size a little bit. And then I take my training data and I generate all those features. And again, this is all running on a Edge Impulse server, so hopefully this will be quite quick. I'm hoping it's going to start. Oh yeah, here we go. And so one of the advantages of doing these models for small devices is that we sort of heard quite a lot about how compute intensive a lot of this machine learning is. If the model that you're developing is going to run on a very small device, that puts a very strong limit on the complexity of the model. And what that typically means is that the learning process is a lot less computationally expensive as well. So when we're training models that are going to run on AudioMoth, those models are going to run on a very low power, very slow processor. So typically we don't need a particularly fast computer to do the training process. And so that's a sort of a key advantage of this sort of tiny ML approach, where we know that we're going to sacrifice a little bit of sort of performance to the classifier, but we're going to get something that's going to run on a very small processor. And so what it's done is it's generated features from that training data. And what it's going to show in a minute is a sort of principal component analysis trying to indicate how complicated this classification task is. And so in a second, it should sort of pop up a little diagram, and we can sort of see the complexity of that diagram is sort of how hard this classification task is going to be. So there's a whole group of sounds up here, which are background, and then there's some sort of more complex set down here, which has sort of a background and a quack sort of mixed together. And then the last stage of this is to train the neural network. And again, we can largely use all of the sort of the default settings that it's chosen. We can sort of go in and tweak those if we really want to sort of change the performance. And then so it's now training the neural network to do that classification task between quack and background. And again, hopefully this will sort of start up pretty quickly, and the process is pretty quick. So you probably can't see this here, but we're sort of training through training epochs. We've got 100 training epochs. We're up to sort of 30 out of 100 at the moment. And our accuracy at the moment is looking to be sort of about sort of 98% or something. So we're about halfway there. And one of the advantages of really nice features about Edge Impulse is that they've done a lot of work with Google and ARM to really sort of optimize the output. So traditionally or previously we'd have used the raw TensorFlow tools to train this model and to test the model. And then we've got this challenge: well, how do we take this large model that we've trained on our desktop and deploy it on a device? And it's specifically that the Edge Impulse tools try to optimize. So try to allow us to train a custom model for our data set, and then optimize that model. And as we see in a minute, the last stage is we can just download that model and then put it into some custom audio mod firmware. So the key thing about all of this tiny ML is that we're really interested in constrained resources. So our device has a slow processor, so we're interested in how long it takes to run the model, and we're also interested in how much memory it takes whilst we're running the model, particularly if we've got a small processor like an audio mod processor, and also we're balancing that against the complexity of the model. And so you can sort of see what we've got here. We've got a little confusion matrix telling us how good our classifier is, and we're at about sort of 96%. You can sort of see some of these sort of confused bits in here. We can sort of go in, we can listen to some of them. A lot of these actual errors are due to sort of misclassification, so we haven't spent a lot of time curating this data set. But at the bottom, we can see that this is a really tiny model. So it uses 10 kilobytes of RAM and 35 kilobytes of storage, so it's very easy to run this on an audio mod. And then the last stage, the sort of stage that we're trying to automate at the moment, is this deployment stage. So what we can do here is we can select a C++ library, we can say build the model, and we can just download that model. And what we're doing is developing the tooling so that we can take that model and then turn it into firmware that we can just run on audio mod. And then what we end up with at the end of that process is a custom firmware that can recognize the quacking frog against the background and only make recordings when it's listening, when it hears the quacking frog. So we then go to the field and we deploy it in the audio mod firmware, and we can only collect recordings then when that noise is playing in the background. So I think this process isn't quite streamlined. It's we need to do some work on our audio mod side to make it a little bit easier to deploy this in reality. The hardware probably needs a little bit of extending. We need ideally audio mod would have a slightly more powerful processor, but we still want to sort of maintain this low power aspect of it. So next generation of audio mod is likely to be a little bit more performant in terms of being able to run these neural networks. But I don't think we're very far away from being able to really sort of deploy this. And we're currently working with a couple of groups. We want to try and develop an algorithm for the plains warbler in Australia, and another one for frogs in Australia. And so I think we're really quite close to the point where, given the constraints of the hardware, so a low sampling rate and quite a small model, we can do species-specific sampling on the device, at least at the level of saying, is this interesting sound or is this just background sound? And that, I think that's a sort of a huge step in this sort of vision of being able to sort of automate the whole process. And so surprisingly, that demo worked quite well. And I've largely used up all my time. So I'll leave it at that. And if anyone wants to ask any questions at the end, I'll be very happy to take questions about how the system works and when it might be sort of more usable by other people. And if anyone's got any particular deployment ideas, we'd be very interested in hearing them.
That was mind-blowingly awesome. So we are running a little bit behind, so maybe just like one or two quick questions if anyone has any in the room or online. No, everyone's had their minds blown. No, there's a couple of.
What sort of minimum? Yeah, just a technical point. What sort of minimum requirements does it have on the processor? What sort of minimum clock speed or memory storage does it require to run this software?
Yeah, so AudioMoth is more constrained than most of the sort of Arduino type platforms that Edge Impulse target. So we end up with we have 48 megahertz processor, ARM Cortex M4, and we have about sort of 20 kilobytes of RAM free, and so we're really constrained. And specifically, so the next version of AudioMoth will sort of address some of those. But if you've got a processor which is running at sort of 80 megahertz, has a few 128 kilobytes of RAM, you can definitely get sort of quite good models running on the device itself.
There we go. Yeah, far away.
Thanks. That's a super cool talk. Like with these kind of deployments, you're likely to care about rare species, maybe more so. So, you know, if you're throwing away everything that's not detection, is there sort of scope to weigh up your recall versus precision in that like platform and say, you know, I want it to err on the side of caution, or is it just one classifier that's like stuck at?
Yeah. So there is one parameter that you can tune. So your sort of your probability of being in either class. Our sort of experience so far is that these very small models aren't great at detecting things when it's a sort of a complex background. But what they are really useful for is just sort of pruning out long periods where there's clearly nothing happening. And those periods are just sort of soaking up your SD card capacity and wasting energy writing to them. And we kind of imagine some of our experience with the sort of gunshot detection is that it tends to be iterative, that you deploy it once and then you come back with lots of sounds of twigs snapping, and then you can sort of go through the training process again, understanding that, okay, well, there's background noise, then there's this class of background noise which is very close to the gunshots, and can we sort of retrain the model to try and sort of distinguish between these two. So I kind of think it's likely to be this sort of iterative approach where you prune out stuff that's clearly background, and then you can gradually sort of refine the model as you get sort of more experience of what typical sounds that are in the environment but actually aren't the thing that you're really sort of interested in.
Okay, we're back in the room, but I think we have to move on, unfortunately. So thank you very much, Alex. It was a great talk. And we've got Simon Gillings of the BTO, who's the principal data scientist at the BTO, who's mainly interested in developing methods and applications that turn BTO data into accessible, robust, and visually appealing products for different audiences and stakeholders. He's currently developing acoustic deep learning methods for bird classification, and is in the room, which is great.
Do you need me to use a mic? Or?
No, I think that might pick you up well enough. Let me just pop your... and then that's the clicker. Hopefully that's working. If not, shout at Alice and she'll click through for you.
Yeah, thank you. Afternoon, everyone, and thanks for the invitation from Ollie and others. So yeah, I'm going to talk about the work we're doing at BTO on a range of taxa. And this is really, as Ollie said in the intro, trying to scale up some of these techniques for citizen science projects that we do. Yeah, that's not working. There we go. So this is our traditional BTO work. Field worker out in the field, counting birds mostly, sometimes other taxa. Oh, it's gone backwards. There we go. But that's not always feasible, as we've heard in a few examples already. We've got landscapes that are hard to access, species that are very rare and you really can't be out there long enough to detect them, or species we just can't hear at all ourselves. So this work we've been doing over the last few years has been really focusing on these kind of monitoring gaps. So we have initially focused on ultrasonic sounds, more recently now working on more of the audible sound range. And we're trying to make these techniques available to our volunteers and other audiences. We're trying to do this in a way that's scalable, so we're needing to use cloud services to make this feasible to deploy across the numbers of users we want to work with, and trying to come up with a sustainable business model for this. So we're a charity, we don't have core funding, so we've got to find a way to cover the costs of running this kind of system. My colleague Stuart Newson, many of you will know, has really started off this work. He's pioneered this work for us, working initially on bats. So Stu identified a gap in monitoring these to his own kind of interests, and has been working on bat classifiers now for 7 or 8 years, I think. He's working on these across the UK initially, but also now increasingly in Europe. And now our system at the moment covers 34 bat species, which is all of the UK ones and quite a lot of European ones, 18 bush crickets, which is all but one of the UK bush crickets, and 14 small mammal species. So I'm not going to say a lot about the actual techniques we've used, because I think they're ones that are familiar to many of you. I should have said at the outset, this particular work is using the Tadorida system, developed by Yves and others, and that's based on random forest classifiers. Of course, the big thing that comes up again and again is getting data and labelling the data, the audio data. So Stu has spent a lot of time going to bat roosts, where there are known bat species, to get labelled data to label, or as here, live trapping small mammals, so he knows what he's actually labelling, because there's a real dearth of recordings for many of these species. My work more recently is using convolutional neural networks, initially focusing on nocturnal bird migration. So this is a time when most people don't want to stay up all night counting redwings, the species here. So I've trained neural nets to scan through these night-long recordings to find redwing calls and other nocturnal migrants. Now that's quite a limited application really, it's just kind of weird people like me that are interested in nocturnal migrants. But more generally applicable, we have interest in the wider community of nocturnal bird species, particularly owls. So these are focuses for quite a few rewilding projects and restoration projects. So I'm working on a classifier for the European owls and nightjars at the moment. But to get all of that off my computer and off Stu's computer, we need to deploy it, and that's what we've developed with what we call the Bto Acoustic Pipeline. Some of you might have seen this or heard of this. So it's supposed to be a public-facing online application. It has 2 main parts, a desktop PC application for handling uploading of the audio recordings and controlling metadata, and that connects to a cloud system in Amazon, where all these classifiers run, sending the results back to you when they're done. To make that work, to make it sustainable, we've gone for this 2-tier system. So if you sign up for this today, you get 100 free credits. And you can, as long as you're prepared to make the data from your uploads freely available for wider use, then you get a kind of a cheap rate. If you're doing commercial work that has confidential, the data are confidential out of it, then you can go for a slightly more expensive rate, and that sort of subsidises this free tier. So we're trying to have a system that allows members of the public, small community groups, to have free access that's subsidised by these kind of bigger projects. And these are some results just based on the use of the system so far this year. So the map on the right shows the locations where this is common pipistrelle recordings. Darker colours are higher levels of reporting rates. So we're already getting kind of distribution information. We've got top left, the range of bat species recorded, dominated by common pips, not surprisingly, and then stuff like hourly patterns. So I think this sort of information is really nice to think about when you need to do surveys for different species having different patterns of activity through the night. So this is these results are quite new, and we want to build more monitoring outputs on top of these, and using these monitoring data to support actual projects. So this is some work we're collaborating with the Endangered Landscapes programme to provide some of the monitoring supports on this range of sites where there's rewilding and habitat recreation and restoration going on. One of the projects we're working on with is in Portugal, and we've been able to confirm the presence of eagle owls at one of these sites, which wasn't known that they were still there. So it's early days for us applying these bird classifiers to these projects, but it's quite exciting to see these classifiers actually finding stuff that's not known about. I'd just like to finish with a couple of challenges. I think we've spent a long time developing these audio libraries, and as many people have said, labelling and collecting recordings, it does require a lot of time and often a lot of personal investment. Developing networks of experts who absolutely know what that particular bat or small mammal sounds like, or have access to a place where you can go and record them, that takes a lot of time. And we want to get these systems used by people who don't have the skills to do that. So building the classifier so that volunteers who don't know how to identify these bats can use a trustworthy system. And as I said, coming up with a sustainable business model is tricky, and it doesn't match some of the other moves for open data, but this is the only way we've found to make this scalable and sustainable in the long term. In terms of issues, a couple of things from our experience is variation in classifier performance, spatial variation as you encounter different confusion species, different novel ambient environments. One of my classifiers works brilliantly until it goes to a place where there are cowbells that sound like a particular owl. Things that you wouldn't really predict sound like an owl. So, being able to understand when your system's not going to translate to a new location or to a new time. Problems at dawn chorus for nocturnal species and stuff like that. I think we need to better understand the performance of these classifiers with respect to detectability. So for our volunteers, we spend loads of time talking about detectability. How far away can you hear the bird? And we talked about how far away can a detector hear a bird, but then you've got this other layer on top of that, is how far away can your classifier classify the thing that your detector has heard some distance away. And I don't think, personally, I don't think I understand that well enough. I don't know how well understood that is for deploying these. And saying, well, when is the best time to do this survey? Because we normally, as humans, will go and do bird surveys at the dawn chorus, because that's the best time to pick up everything. But it might not be the best time if you're trying to detect things in dawn chorus recordings. Maybe if we use occupancy modelling and understand the detection processes, maybe it's better to record at a different time of day and to really understand the detection process better. So yeah, what's missing? So I think understanding more about detection distances, interspecific detectability for these systems. I've heard a bit about recorders. Is it better to have one expensive recorder that has higher detectability, or a network of 5 that can space closer together because they're cheaper? And ultimately moving from just detecting presence, absence to getting some measures of abundance so we can measure trends more accurately. So, part of this is about these generic guidelines. What do we need to compromise on? Our volunteer surveys cover 100 breeding bird species, but then those methods aren't perfect for any one species. It's a compromise that works well enough for long-term monitoring. And I'm wondering, what are the compromises we can live with that give us the answers we need with sufficient precision? So I'll just finish with this last slide. So thanks to Stu and Adam, who's in the audience as well, who's leads on this Endangered Landscapes programme. And if you've not seen it, do have a look at the Acoustic Pipeline. Thank you.
Thank you very much. Thank you very much. That was great. And seeing as you're in the room, I can do the running microphone around this time. If anyone has any questions. Anything online?
Thank you, Simon. That was excellent. The questions rather predictable considering what I spoke this morning. The pipeline, I'm just busy trying to find it on here just now. The data, is it DOI'd or how is the data stored? Can you tell us a little bit more about that, please?
Yeah. So at the moment, the data are all stored well, there's two sorts of data. There's the original recordings, which are all stored in deep blast storage in Amazon, because it's the cheapest place to store them. The kind of detections, so the species IDs are all in a database. You can visualize them, like I've shown. They will be available for people to access, but we've got there's an issue about validation, which I think is something else we need to talk about. Is we can build a system that has very high accuracy, but people at the end of the day can still misuse that, expecting everything to be absolutely perfect. You know, 98% accuracy when you've got 9 million recordings, that's still a lot of errors. You know, so we haven't yet made all those data available. We want to build a verification tool on top of this, so that we can get some of the recordings where it really matters, get them checked a bit more thoroughly. But we do want to make the data used. That's why we've set it up with this shareable setting. But it's just this, I think, particularly for bats, where there's such a lot of politics, there's a lot of sensitivities about releasing locations of roost sites. We're just being a bit cautious, but we want the data to be used ultimately.
Kahl? Yeah. I was just wondering about the upload to the pipeline. Is it still true that it has to be 192 kilohertz sample rate, or can you put in lower sample rate data?
I think it does have, yes. We're not. It's still focused on the ultrasonic species. So when we get the bird classifiers in there, I think there'll be sort of 2 routes.
Anything online, Alice?
No, not apparently.
Any others in the room? One over here.
Good job.
Simon, great talk. Just wondering, what are your thoughts and ideas for estimating relative abundance from acoustic data? You mentioned it in the last slide. What are your thoughts on that?
Yeah, so I know there's developments in the tech to have pairs of recorders that are synced and other ways of potentially localizing to get more of a sort of 3D soundscape that would provide some measures of abundance. I guess it really depends on the question. You know, is this about getting site information for the site, an inventory, absolute abundance, or is it for looking at long-term changes in relative abundance? I think that's a critical question for all of these guidelines, is, you know, which of those two things are you trying to answer? I think, and again, it depends on the taxa. So, you know, if you put, for these bush crickets, you get millions of recordings of the same bush cricket. You know, it really doesn't tell you a lot. So I think we need to get this multiple detectors probably, and then ideally some ground truthing of the recorders and some other measures, independent measures of abundance. But we don't have that for the bird data we're working with yet.
Do you think there's opportunities here for using things like N mixture models, which would give you estimates of detectability and relative abundance, and just develop sort of acoustic capture, recapture? You don't need to, with N mixture models, you don't need to recognise individuals.
Potentially, I think understanding how to build in the uncertainty of the species IDs into those,
Yeah.
So you don't, you know, those models normally assume truth is 100%, but they, you know, so there's uncertainties got to go in at the first level of that.
There's occupancy models now, aren't there, that build in uncertainties.
Brilliant. Thank you very much.
Okay.
Great talk. Okay, so I don't have to fiddle with sound levels too much for another 10 minutes. We'll have Carlos Abraham, seeing as he's in the room. And
Carlos, is it still on your pen drive?
I opened it. It should be open.
Well, it's open again now.
That's it. Okay, have it twice. Let's have it. Capicalia rather than Alex on the screen.
Well, it should be you, but I don't know why it's not. I think that's okay. Yeah, have you got the clipper?
I've got a clipper.
Excellent. Okay, you're good to go, I think.
Okay, hello everybody again. I'm going to dumb down from machine learning and complicated things and talk about some work I did a few years ago on this beast, which is the Capercaillie. I imagine some people will know what those are. For those who don't know what Capercaillie is, it's a big black turkey that lives in the woods in Scotland, basically, and is currently going extinct in the UK for the second time at the moment. Went extinct 150, 200 years ago and was reintroduced, and is not doing very well because of climate change, deer fences, and those sorts of things. And back in 2015, 2016, did some work with the Forestry Commission, RSPB, up in Scotland to see whether we could use acoustic approaches to monitor this species and see how it was doing in its breeding grounds in the forests. Cool. We've shown these spectrograms yet? We've shown a couple, haven't we? Not everybody in the room perhaps has not used spectrograms before. I thought I'd show one of these to start off with. But when we're analysing sound data, and most of you will know this, yeah, we can use spectrograms to identify species. We do this for bats all the time, consultants like me and others in the room, but we can do it for a range of other species as well. And yeah, we've got some great tits and chipchaffs and chaffinch and blackcap and stuff like that. So we can record the sound, listen to it when we take it back to the lab, or view it on screen and use that to identify particular species on a manual level rather than doing machine learning. A lot of the machine learning approaches do the same as this, where they produce a spectrogram and use image matching to identify species as well. We've done this already, but back in 2016 I used SM2 units, and in 2017 again we did 2 years of research up there on Capercaillies, and recorded the sound using remote units that were based at the leks. So Capercaillies have a lekking mating system where basically all the males gather around to have a bit of a dance-off in the middle of the forest in Maytime, trying to impress the females, and then the biggest, loudest, showiest, heaviest male normally wins the day and gets to mate with all the females. I'll skip that slide. So basically collected data using a fairly simple survey design. We basically already knew where the leks were from the RSPB research work that had been done for years, and basically the RSPB have been monitoring with their other partners, Forestry Commission and National Trust, monitoring Capercaillie leks in the forest, and they know where they all are and monitor them using 2 different systems. And we wanted to try and see if we could develop a workflow where we put acoustic recorders out and compared the results that we got from these traditional techniques with acoustic techniques and see how the results compared from that. And in terms of the workflow for the manual processing, it was some supervised approaches using Kaleidoscope Pro software. So the Wildlife Acoustics software has got an algorithm built within it where you can do a clustering analysis. So it's sort of halfway towards machine learning, but you don't need a training set. So you're not getting known labeled data and matching new data to that. You basically just feed a load of data in, and using magic, which I don't understand, it looks at all of the different sounds. So it detects sound events, little chunks of sound there, and it compares them all with each other and puts them into groups. So you end up with group A, or cluster 00, cluster 01, cluster 2, and whatever else. And when you look at that data, you should come out with different sorts of calls within those clusters, or project into multidimensional hyperspace or something like that. It works to a certain extent that when you do this with bird data, you tend to get lots of blackbirds in cluster 0, yeah, some different blackbirds in cluster 1, but then lots of blue tits, grey tits, and it does sort it out pretty well into different species. And so basically took that approach to analysis from this, where we stuck the records in the field for about 2 weeks the first year, and then for a full month the second year. We did only 4 LECs in 2016, but went back and did 10 different LECs in 2017. So a little pilot study to start with and a later study, and analysed the data using this clustering process to try and identify Kappa-Cayley calls, these segments, using this semi-supervised thing where we did let clustering do its thing, and then lots of manual checking at the end to see how well that had worked and pick up on all the faults and negatives and the false positives in the data. And so we ended up with lots of Kappa-Cayley calls from that process. I won't try playing the sound because we've got enough issues with sound today. But Kappa-Cayley's had a pretty distinctive song, if we can call it that. They start off with this sort of lowish frequency drum roll, lower frequency than that, and then the cork pop note, so a single, and then they have this wetting phase, as it's called, sort of a scratching thing bit at the end. And that bit's not particularly distinctive, but this bit is. And the cork pop note is really important for Kappa-Cayley breeding, and one of my favourite papers ever in northern Spain looked at recordings from individual Kappa-Cayleys with a gun mic, recorded Kappa-Cayley A and Kappa-Cayley B, the males, looked at the frequency of the call that they produced, and that was related to the size and the fitness of these males. Basically, the fitter that the birds were in breeding terms, the lower frequency the note was. But as well as doing that, they looked at the quality of the habitat around the lakes that these birds were using and the overall bird assemblage that was present there as well, and found that the bigger, heavier, sexier males with the lower frequency notes lived in the best habitat, and that best habitat supports the greatest species richness in terms of the overall bird assemblage. So you can actually relate the frequency of that cork pop note to the overall bird assemblage, at least in their paper they did anyway, and use that to assess, the breeding bird assemblage just from one note like that, and how many kilohertz it is. Anyway, it's a fancy bird with a nice call, and we recorded lots of them. The standard LEC monitoring methods, or the methods for monitoring Kappa-Cayleys in Scotland, are twofold. There is either going to the LEC during the breeding season, which is sort of late April, May sort of time, when all the males gather together. But because these are very disturbance-prone species and they're listed under the Wild Countryside Act schedules, you need a survey licence to go and record them, and you need to be very careful about not disturbing the birds. So basically the surveyors have to go to site the day before, put a tent up they're going to use as a hide, go away, come back under the cover of darkness, sneak into the tent at sort of 11 p.m, wake up at 3 p.m, stick their heads out, and then try and record how many birds there are going in the woods around them to work out how many males are using the lake. So not easy, and not easy to cover a large landscape across Scotland to try and record those things. So not very. It's a good survey method, but hard to implement across big landscapes. The other survey method is not to bother with recording them at the lakes, but try and find the species by walking around in Scotland in the forests and trying to bump into male or female lakes as you're doing that. The average success rate for doing that is, in a Scottish winter, in the forest, is one bird every 22 kilometres. So hats off to the people who do it. It's used both of these methods together for the national monitoring for this species, but hard work, and so if we can just automate it using acoustic recorders, then why would you not? So that's what we were trying to do and see whether we did that. And a couple of papers that we did, and they're out to publish a few years ago if you want to read them. So we established that you could record Kappa-Cayley the first year, and we could get good data from using acoustic recorders, as have been found but a couple of other places as well. We recorded for a whole month in 2017, and this is the numbers of Kappa Cayley calls recorded per day through the course of that month, and you can see it's not very consistent. It goes up and down and up and down and up and down. Mostly, depending on the weather. If it's windy and rainy, yeah, the birds shut up and the birds are harder to record, so you tend to get less numbers of calls out. These are basically the two same things, but just expressed slightly differently, and I probably won't go into the differences in there. But so you can record the birds, but what days you pick to do them will give you differences in the numbers of calls that you're getting there, which may be related to the numbers of birds that are on the site, or may not be, depending. We could track, because we've got high resolution quantitative data for how many calls we've got per hour at each of these sites. We can see that this huge tidal wave of Kappa Cayley local activity occurs just before sunrise, which is this line here, fades off during the day. We can also see that these 4 different sites from 2016 data have very different times in terms of when the Kappa Cayleys are actually doing their thing. And there's some anecdotal evidence that in some of these sites in Scotland, where they get lots of dog walkers around, potentially the Kappas come out and do their displays before the dog walkers come out at 7 AM. And on those sites, they think that they do their display a little bit earlier rather than a bit later to avoid disturbance from recreational things. Also, they may vary the time depending on aspect, altitude, and that sort of thing, and sort of, as you'd expect in terms of sort of sunlight coming into the sites and how quickly they warm up, etc., etc. You can tell that from the data. One thing that we wanted to try and work out was whether we can use very, very simple maths, yeah, not occupancy modelling or anything else, to see if we can record the numbers of calls that we're recording per site and relate that to the numbers of birds. If you're interested in that, then you want to look at the papers of Christian Perez-Granados, who's done a fair bit of work on local activity rates in different species, and other people have done stuff on seabird colonies and penguins and that sort of thing as well, which show that actually just the volume and the variety of calls you get within a certain time period is a good reflection on how many birds are present, if the birds are close together and in a mass, like in a seabird colony, or at a lekking site like this one potentially. And we found out that for all these different sites, basically the numbers of males recorded from the people sticking their heads out of the tents at night time were reasonably well related to the numbers of calls that we got. So we can actually use the numbers of calls, this call index, to identify to some degree of accuracy how many birds there are. And in fact, you could flip this the other way around, really, because I'm not quite sure whether these numbers down the bottom are correct here or not, because, as I said before, it's a bit hard to tell whether you've got 5 or 6 Kappa Cayley in the forest in the middle of spring nighttime as well. So there's probably variety on both axes there in terms of how accurate these things are. One thing that we didn't expect to find in the data, and this was a bit of a chance occurrence, was that we know at each of these lecks the numbers of calls varies from day to day. And we had just a spurious sideways look at what happened when the people turned up to do their lek counts, which is where these red arrows are. And if you see at some sites, like this one I'm pointing to here, yeah, fair bit of activity, but nothing, not very much, nothing, and etc., etc. And so it would appear, and if you graph this out, again, there's a significant relationship between the presence of surveyors. So it seems to shut up a bit or clear off, or both, whenever the surveyors turn up. And so we need to think about how good is the lek count when they're potentially causing disturbance on the birds they're trying to survey. How we do for time? Probably there. Yeah. Good. I won't talk about occupancy modelling the Thames Basin piece for Nightjar, but if anyone wants to speak to me later about that, they can do. So.
That was a really good really applied, interesting talk, I think. Does anyone? Yeah. Does anyone in the room or online have one or two very quick questions? No, online.
Really quick, why not leave a camera to watch a letting site and just count the number of birds?
That has been done, and both methods have been done together. So there's some guys in Austria, Switzerland, Italy, Spain who took different approaches to that. And it depends whether the lek is in where you expect it to be when you put a camera out, for one thing, and it's quite a focused beam. You'd have to deploy potentially quite a number of cameras to cover the lek site, because black grouse, similar species, tend to have quite a small lek site, but within Capricalis they're dispersed over 50 or 100 metres away from each other, potentially in an exploded lek, as it's termed, so one camera wouldn't do it.
And it's very data intensive as well.
One more very quick question. Anything online, Alice?
No.
No. Not very inquisitive lot online, so.
You did miss one earlier.
I'm not sure if it would work, but is there an application for surround sound systems in this? As in to record surround sound and then do the count afterwards, just using units.
Potentially, for Becky was talking about time of arrival differences at location, and lekking birds would be a prime use for that, because we're in fairly constrained. We do that with 4 or 5 echoes over sort of 50 or 100 metre distance, which is probably about decent size rate for a Capricalia lek.
Hi, Mike. No. Why? Okay.
We are, yes. So next up we have.
Right.
Oh, we could do David. Let's do David, seeing he's here.
Was it not supposed to be me?
Yeah.
Are you ready, David, or should we come back to you?
Ready, yeah.
Okay. Yes, you're muted because I haven't got the sound sorted. But yes, so David Jarrett is a PhD student at the University of Durham using acoustics to monitor breeding waders. He's got a really cool study set up in northern Scotland, middle of Scotland.
Northeast, yeah.
So I will put the sound on. If you can screen share, David, as well.
Yeah.
Then ready to mute.
Can you see that? Brilliant. Okay, so I'm just going to talk briefly about my study using audio moths acoustic monitoring to better understand breeding waders. And I'm at my study site in northeast Scotland at the moment in Aberdeenshire. This work is kind of, I'm doing my fieldwork right now, so I haven't got too much analysis or results, but I'm going to talk about the design of the study a little bit, because I think that's really interesting and kind of a really important part of acoustics as well, or bioacoustics as well. So the background and the rationale for using acoustic recorders to monitor breeding waders is really interesting. So we know that waders are declining really rapidly across Europe and other parts of the northern hemisphere as well. And certainly in Europe, low breeding productivity is the proximate cause. That's in some places that's related to agricultural activities, other places generalist predators and other types of disturbance. It's really difficult and labour intensive to gather good data on breeding productivity using survey, as waders leave the nest sites within about a day of hatching, then they're out there in the world. And when a surveyor walks into a site, the adult birds tell the young birds to lie down on the ground and don't move, and so it's difficult to see them, and it's difficult to count them, and it's difficult to determine whether a pair has young. But there are some cues that you can use. Added to that, we have lots of waders breed in northern latitudes where survey axis is really challenging, and northern tundra landscapes are full of breeding waders, we generally don't have good ways of
David, you've muted yourself. David, you've muted yourself.
Can you hear me now? Hello? Am I still muted?
We can hear you.
Oh, sorry. Where did you lose me? I'll just carry on anyway. So as I said, lots of waders breeding in northern latitudes where it's difficult to access, and we also have in agricultural landscapes we have really poor outcomes from agri-environment interventions. And again, that's limited to not knowing too much about where birds are breeding productively, where they're not breeding productively, because of the limitation, because of the difficulties of gathering productivity data from field visits. So there's this really big need for good enough productivity monitoring. It doesn't have to be perfect, but actually a kind of an approach that we could roll out across a large scale to gather some data would be really useful for lots of purposes. And as I said about productivity surveys, a really important part of productivity surveys is audio cues. So some waders have specific calls which they make which they only make when they have dependent young, and recording these particular calls is how we kind of gather data on productivity from field visits. And then furthermore, waders are really most species of wader are really conducive to acoustic survey designs because they're loud, and the calls often carry across large distances. They also breed in species poor habitats or environments, which is also really useful because relative to some other challenges, it's actually the challenge of sifting through data to pick out your target species is actually not really much of a challenge with some of the species I'm talking about. Okay, so the work I'm doing is sort of separated into a few different strands. So the first thing, as I said, really interested in breeding productivity, and this question of whether you can use audio moths, so deploying audio moths in the breeding grounds. Can you infer whether birds are breeding productively just from the vocalization patterns that you gather from the audio recorders? And the way we might do that with a bird like curlew, for example, if I play this second call here, this second call, not this bit. So that call is almost only used when the birds have dependent chicks. They will use it in certain occasions when a mammalian predator is very close to the nest before the chicks hatch. But by and large, they only use this once the chicks hatch, and certainly if a human walks through a breeding area, you hear lots and lots of this call if there are dependent chicks in the area. That one. So then what I did last year was deployed audio moths in various areas where I expected particular productivity outcomes. So the site at the top, we had high chick survival and high productivity, and the graphs at the bottom describe a low productivity site, and we can see that there's quite a strong difference. So the graphs are describing precisely the same period. So at these sites, egg laying was somewhere between the 15th and the 20th of April for most pairs. And you can see that at the site with low chick survival, the graph at the bottom, vocal activity attenuates quite quickly before such time as birds were reared to fledging, whereas at the site where chick survival was high, vocal activity continues for quite a lot longer. The data on alarm calling is a bit different. It's probably a bit confounded by the fact that the low productivity sites had high levels of mammalian predators, and that means you get more alarm calling. But again, the alarm calling attenuates a little bit earlier as the chicks disappear. But this is kind of confounded probably by the effect of relaying, and so when birds lose eggs, at least they'll relay, and so you'll have further breeding attempts. But certainly across all of the vocal activity, you can see that there is this effect where you get a different vocalization pattern at high productivity sites compared to low productivity sites. So that gives us some information. I'm also really interested in whether acoustic monitoring can produce good information on relative density or relative abundance. I think it's been mentioned a few times already. And again, curlew are a really good bird to do this with, because their calls travel a long distance. It's not like searching for kind of difficult to detect birds. I think if your target species is quiet, it doesn't vocalize much, the song doesn't carry very far, then it's going to be really, really difficult, because you're just going to have to have a really, really large sample. But with curlew, I think it's probably more feasible. So this is where I am now. That's my I'm actually in this little house there, and this is broadly speaking, this is my study site. It's about 30 square kilometers in the east side of the Cairngorms, just in Aberdeenshire. And this is a map of the site. So I'm walking these transects using sort of standard BBS type methodology and then distance sampling to characterize the densities of different bird species across the estate, but with a kind of a focus on curlew. And I'll model the relative abundance using kind of sensible variables like habitat, elevation, and slope to produce a kind of density surface model of particularly curlew, but other species as well. And I've got a whole load of audio moths. The green dots are standard audio moths, and the pink dots are GPS synced audio moths, which I'll talk about in a second. They just recording in 5-minute chunks across the sort of the morning, really, and I'm intending to use the percentage of each 5-minute period occupied with each species vocalization as a crude index of abundance at each point. That kind of vacant blob has been filled in the last few weeks with more audio moths. I don't know how many dots there are there, but I don't have that many audio moths, so I am moving them around during the breeding season. So that will then be used. Obviously, I'll try and do a kind of similar spatial model using the acoustic data I get here, and compare that to the spatial model that I can produce from kind of standard distance sampling transects. And then, yeah, the last thing is sound localization. So I've got some audio moths where I can sync where we can sync the clock really accurately, and therefore use them hopefully to localize sounds within the grid. So just for an example, here's a curlew nest, and what I would ideally like to do would be able to locate nest sites within my grid. And so that call there is very characteristic of a nest which has just hatched. So the curlew on the nest is calling her chicks back to be brooded, I think, or she's contacting them somehow. But this call is really frequent when just after a nest hatches. So ideally, what I'll be doing is trying to localize these calls. There are one or two other diagnostic calls like that within my grid to see if I can get clusters which are indicative of nest sites. So I know where all the nests are within the grid as well, which is obviously useful. The other thing I should quickly say is that I will also be using the gridded audio, the GPS synced audio moths to generate detection curve. So just taking vocalizations, whether they be alarm calls or display calls or whatever within the grid, I'll know whether they're recorded I'll know with what probability they're recorded at each distance, based on whether they register at the various audio moths. Yeah, I think it covers all that. And that picture in the bottom left, that's the one in the middle. Basically, that's to show that we're kind of we're recording omnidirectionally in a very crude fashion. And then just the last little bit. I mean, I sort of know this already, but I've got. This is a curlew nest, so here we've got cameras on these nests, so we know when they hatch exactly, and then the audio moth at the nest will also just give me a bit more data on maybe how frequently those calls are made at hatching, and also whether there's specific calls made at incubation. The nests tend to be pretty quiet during the incubation period, understandably, but there may also be diagnostic calls which we can use to identify nests during the incubation period as well. Yeah, that's it. Thanks for listening, and thanks for inviting me. And I'm sorry I couldn't come down. I would have really liked to come down, but I'm sort of busy with audio moths at the minute. Just out of interest, anyone wants to contact me, please do so, and I'd be more than happy to come down and talk in more detail anywhere really about this work in sort of the autumn or winter when I've got more results and data to talk about.
Thanks very much. That was again a really interesting applied approach. We are running super late. Still got another 2 talks in this session, and we should be finishing the session now. So if anyone's got a really quick question, or if not, we'll move straight on. Okay, no one's been quick enough. That's great. One person's quick, gotten there quickly.
It relates to something that you were talking about already, Carlos, which was the use of call activity to indicate abundance. I was wondering if you could elaborate on that and whether that's something you can use sort of across the board.
Yeah, I can chip in.
The simplest method to assess abundance from call data is the one I've used, which is basically how many calls in a day or an hour or whatever, and is that related to the numbers of birds that are making that sound. That's been shown, as I said, for seabird colonies, lek sites, but also flocks of some birds, like bee eaters and other things that they've looked at. The other approach is to try and use acoustics to understand the geography of the birds, and using triangulation from different recorders to spot where the bird is and use that. Or you can use occupancy modelling methods, which doesn't give you abundance, but it gives you some measure of density and territory size and that sort of thing. So there's a range of different approaches of complexity. Some of the time, yeah.
David, do you have any thoughts on that, just quickly?
Yeah, no, I mean, I totally agree. I think I'll have data on how many calls at each recorder and also what proportion of time is occupied by a vocalization, and I'll be able to see which one correlates better with the data from the transects. I think that one of the challenges with curlew is that their territories can be 2, 3, 4 square kilometers depending on habitat, and so it's a little bit harder in that what the recorders are doing, but also what the transects are doing is just getting a relative density of vocal activity at a particular point, and it's not so much, it's not quite so easy to say, well, this site is occupied and this site isn't, because birds just move around a lot in the landscape, and there'll be some areas where you get clusters of birds that are active but not breeding, or vice versa.
Brilliant. Thank you very much, and thanks for that, David. That's fantastic. Just quick on the admin, we're running quite late, so what we'll do is we'll push the parallel session back by 20 minutes, we'll push the poster session back by 20 minutes, and we'll end up in the pub 20 minutes later at the end of the day. But it'll be okay. We'll be okay. There's some wine with the poster sessions. You'll all survive. David, if you could turn your camera off now. And yes, next up we have two fantastic eco-acousticians from the other side of the channel who both work at the French Natural History Museum, but in different research groups. And we're going to start with Yves, who also, like Simon, well, Yves works with the Tadarida software and with citizen science.
Thank you. Can you hear me? Okay, I suppose so, because I can't see anything now. Okay, please tell me if there is anything wrong about the visual or hearing. So thank you for the introduction already. I will talk about what I'm doing with quite a lot of colleagues about acoustic monitoring, mainly on bats, but for the same reason as Simon Gillings told before, we are also targeting other species, and in fact we worked quite a lot with Stuart Newson at the beginning of the project. So just quick figure on the main terrestrial groups that are using acoustics for long-range communication, and you see that there are a few groups using ultrasound, so a few groups that you need technology to monitor them. You can't, there is no traditional way to detect them, really. So bats, obviously, but also bush crickets, like Simon said, because they are using the same frequency, and they are active during night and summer. So when you're targeting bats, you basically get a lot of... oh, it doesn't work. Ah. Okay, I thought I was sharing it. So should I... Okay. I'll try again. Is it good right now? No, it doesn't seem so. Yes, it is. Okay. Okay, I guess it's okay. Please tell me, because I don't see Teams when I'm going in the present view. Yeah, it's okay. Okay, I imagine so. So we have been proposing 3 different protocols. So VGKIRO, which is a monitoring bat population monitoring scheme in France based on acoustics, started in 2006 with acoustic recordings, so automatically triggered but manually analyzed with 2 protocols, so car transects and walk transects. And then in 2014, we proposed a 3rd protocol because there was a new technology that it was possible to record ultrasounds during full nights at this time. And we proposed the 3rd protocol with full night stationary recordings. The 2 oldest protocols, car transect and road transects, have a moderate geographical coverage with 200 sites each, and but full night recordings have been really popular lately, and we are reaching 15,000 points sampled in France, with an important proportion that have been repeated several years. And on this data, we are now able to calculate the trends of 6 species of bats. on which 4 are declining, some really strongly, and it is feeding a national indicator, BAT indicator, that synthesizes all those common BAT trends, which is declining by roughly 50% during this period. For now, the full night protocol, which seems to have a very high potential, has collected only a short time series. Like you said, it's only in the 3-4 last years that we have more than 10,000 nights recorded per year, and at the beginning there was only much fewer. So these time series are basically on average like 4 years, 5 years, no, not more. But still we have some trends, not very robust, but consistent with roost monitoring. And we have also, I skipped that, but we have also significant trends for bush crickets, for 4 declining and 1 increasing. We are also using this data for dynamic mapping, so to compute prediction on the whole country. And Charlotte Cromer is working on a project on migration. So this is the seasonal predicted activity of liceless bat, which is a migrating bat. So if you may be able to see the flash in September when the bat is crossing the eastern valleys in the north-south axis. So just that was a quick overview of the program and the data. I'll talk a little bit about what we are doing about automatic identification, so why we are using it. So maybe it's obvious today, but at the beginning we were manually checking everything. So it was a few tens of thousands of recordings each year. So it was time consuming, but obviously it's not possible today because we have like around 10 million bat passes recorded per year. So some colleagues started to do machine learning development more than 10 years ago. And it was implemented in 2015 in a web portal. So that the monitoring data is processed automatically when it's uploaded by the participants. So now we can process the data thanks to automatic ID. There are some good stuff with automatic ID that is not so well known, is that it can control observer bias, and it's quite important for long-term monitoring programs because participants, humans, tend to increase or at least to improve their identification ability over time, and it can produce quite a lot of bias, and it's the case for bats, where we know that we did not have the same knowledge 15 years ago than we have today. So to be able to reanalyze all the historical data with machine learning make us able to control this observer bias. And the other good thing is to be able to get data from the non-targeted taxa, on which not so many people are interested in, like the bush crickets, for example. And we work with Stuart Newson on that. So a quick overview of the Tadarida software. It's a modular software with sound event detection, like anyway, extracting quite a lot of features in the first version. The second box is about labeling. So you heard about that. That's quite important to be able to label a lot of data. And the original thing with Tadarida is that you are labeling pre-detected events, so you don't have to segment yourself. So at first we used mainly random forest algorithm actually to train the model, to train the classifier, and we trained it in a kind of active learning, basic active learning process by feeding it with the newly collected data from the monitoring scheme. So having a lot of data coming from a monitoring scheme make you able to do a stratified random sampling of your data and oversample errors or quite atypical situation, or you maximize the diversity of your training data. So inputting each time with this kind of loop, you will obtain a better fit, a better performance on the at least on your data set. So obviously, so when you train it, so you can apply it to any new data. So that's what we are doing. And something we worked a little bit. We worked with Oliver, and we are implementing it right now on our web portal, is contextual classification, and that's quite important. Is that most of the audio classification today is not taking in advantage of the fact that when you have long time recordings, you have a lot of information that can allow you to correct errors according to context. So what we are using is basically what has been obtained by a Yves classifier in terms of the scores of each species and the ratio of the number of detection between species, and this decreased a lot the error rate for bats and bush crickets. And this is fed by a lot of collaborative checkings among participants, and we provided the Shiny app to do for the participants to stream collaborative checkings. So it's a Shiny app when you can see the detection, the history of detection of bats or on bush crickets during the night. So you have time on the x-axis and frequency on the y-axis. You can do a lot of filters, and you can get the file path by clicking on the point where you think there is something interesting or weird, or I don't know, that needs to be checked, and then you can confirm or correcting the ID. So all of it is open source. It is for the participants. It's available through a web portal, like I said. So it's basically people upload the data through the web. It is automatically and securely stored in IN2P3. So it's a National Institute for Particle and Nuclear Physics in France, and they have huge facilities and subsidies to make it available for biologists. So we are quite lucky with that, and it is relatively low carbon footprints because the definitive the long-term storage is made on magnetic bands. So it is stored, and obviously it is also processed, and participants get an automatic feedback, and like I said, are able to do some manual checking. We are now involved in the development, like many people, many of you, of the use of deep learning, and not for bats, but for the audible range, because we are also recording quite regularly nocturnal birds, frogs, and other insects like ground crickets. And at least for birds, it seems really, really interesting to use deep learning instead of random forest. So like Kate Jones said this morning, this is keeping the feature extraction step. So we are classifying on the spectrogram itself. And it sounds promising, still under development, but it's open source. So if you want to take a look, you can. A brief word, if I have still a little bit of time, about how do we deal with error. So it's a common features of automatic ID, of any identification. You have errors, but for automatic ID, you have something else. You have errors, but you have also confidence of the classification. So we worked quite a lot with colleagues on Kevin Barry, on this Kevin Barry's paper, on the correlation with success probability of the identification and the score given by the classifier. So you see, like, there is a dozen of bat species here, and the logistic regression are showing the relation between those two. And basically, when you have a steep slope for most of the species, it means that for the highest score, you have like a large range of score when you have a negligible false positive rate. So if you push your threshold, relatively careful threshold, you can get pretty clean data, or at least very few false positives. Interesting case here, briefly, is the Pipistrellus natusi here is pretty bad, because at the best score, you still have like 30% risk of error. And this is basically, this is not really because this species is not easy to identify, but it is because it is much rarer than its most closely acoustically related, I'd say, Pipistrellus cooley. So this is something very important in automatic classification, is that it works relatively rapidly well on common species, but for the rarer, for those who are close to commoner species, it is pretty difficult. You need a very, very high performance to be able to get good data. We also, last point, we also try to propose a method about controlling this bias. So obviously, when you have a species where you have a lot of false positives, this false positive rate is biased by the abundance of the other species. So so you can try to reduce so quite often false positive rate is more costly for ecological inference. So you would be tempted to push the threshold so that the false positive tend to be low, but the false negatives are not always randomly distributed in space. They are usually induced by low quality calls, and this can be induced, for example, by vegetation density or something. So you can also have bias with a very careful threshold. So our proposition is to repeat the analysis with different threshold and check for the consistency of your inference. And that's what Kevin did on this study on the effect of distance of wind turbines on the bat activity. And on this very specific gradient, the estimates very, very little. And we also replicated this method on other study on artificial light, motorways, and so on. So in conclusion, I think we can say that large-scale ecoacoustic can work, and the combination of thematic ID and citizen science and providing this freely this auto ID through web service is quite productive. And also that generic sound event detection, making it the most generic possibly, make you able to target quite a large diversity of taxa, so that allow us to share cost and benefits. And for prospects, yeah, like many of you already said, we have many bias in acoustic data to investigate, like masking, attenuation, and so on. And of course, we are still have a large room for improving automatic ID. And obviously, we still need more reference data, especially for everything else than bats and birds, I'd say. And we are currently, like many others, trying to add audible frequency in the process, so that for the very same monitoring scheme, we can also compute trends for nocturnal birds, frogs, and ground crickets. So thank you for your attention.
Thank you very much, Yves. I think we've completely run out of time for questions, I'm afraid. But if you could monitor the Slack, Yves, and hopefully people can put questions into that Slack channel for you. Okay. Yeah, I'm really sorry to just overrun. And we still have one more talk from Sylvain Haupert, who's a great data scientist, sort of acoustic engineer, and he's going to be talking about something that's come up a few times today already, and that's sort of detection distances in acoustics. So over to you, Sylvain.
Okay.
Is everything okay for you? It's terrible, I cannot see anything. So, hello, thank you very much for giving me the possibility to talk about our research. Today I will show you that the detection distance of autonomous recorder units varies over the dial cycle and across seasons. This this might have an impact on species-specific monitoring as well as in soundscape monitoring. And this work was done at the Muséum National d'Histoire Naturelle with Jérôme Sueur and in collaboration with Frédéric Seb. So global warming is already changing the soundscape of our forests. So ecoacoustics seems to be an appropriate tool to track the changes of the biodiversity. And from this constatation, we started in 2018 and 2019 two long-term audio monitoring projects in two very different ecosystems in France and in French Guiana. The goal is to wiretapping both forests for at least 15 years. And today I'm going to focus on the Jura for this presentation, but we did the same experiment in the French Guiana. The forest is located near the Switzerland border in the Parc Naturel Régional du Haut-Jura. The name of the forest is La Forêt du Rizou, which is a Natura 2000 site, and we call that project DB at Rizou. The ecosystem of the Rizou forest is an alpine coniferous forest with poor spruce patches mixed with beeches. And when we started the project, we wondered how many recorders do we need and where do we place them. So this question is essential to define the number and position of recorders and is linked to the listening distance of the recorders. And the listening distance is also required to provide reliable estimation of species richness, abundance, or occupancy, as all these indicators are per unit area. And here, for instance, we have in orange the listening area of a recorder, and we can see that is able to listen to 4 birds. And in some cases, the listening distance can vary. We will see that later. And in this example, the radius increases such that the new listening area is now the green area, and the number of birds that can be detected by the same recorder is now 13. But when the area is taken into account, the density of birds remains the same as when the listening area was orange. So now you understand why it's so important to estimate the listening distance of the audio recorder. The listening distance depends on 4 factors. The characteristics of the receiver, which is the audio recorder, and the characteristics of the emitter, for instance, a bird or a frog. And the characteristics are its position and orientation, but also the sound level of the call and the frequency content of the call. A louder call propagates farther, so low frequency call also, while higher frequency call or faint call propagate less far. We need 2 more factors that are usually overlooked to understand what's going on. One of these factors is the transmission channel. Sound does not propagate in the same way in forests, which are closed habitats, than in grassland, which are open habitat. And the last parameters, it's the ambient sound, which correspond to the sounds produced by biotic and also abiotic sources. The ambient sound is often called background noise because we don't want it. In communication process, the background noise is the sound that masks the signals of interest. So the objective of this study was first to find how far a recorder is able to capture a sound. And this can be reformulated in another question, which is at which distance a sound is below the ambient noise. To answer these questions, we require an estimation of the outdoor sound propagation attenuation. And this outdoor sound propagation attenuation is composed of 3 attenuations: the spherical dispersion or geometric attenuation, and the absorption, the atmospheric absorption, sorry. Both can be estimated from the theory. We don't need to evaluate them. And the third attenuation is the attenuation due to habitat, and this has to be estimated experimentally. It also requires the estimation of the ambient noise, and this will also be estimated experimentally. The experimental protocol involves an audio recorder attached on a tree. In this case, it was an SM4 and a loudspeaker. The loudspeaker faces the recorder at the same elevation, about 2.5 meters, and we move the loudspeaker from 10 meters to 100 meters on the transect, and at each position, we record 22 seconds of the ambient noise, or ambient sound, followed by 22 seconds of white noise. So the results is presented on this graph, on this slide. On the graph here, you can see the spectrogram of the entire experiment, which is a sequence of ambient noise. We just listen to the sound of the forest, followed by a white noise signal that is propagate, and so on. And so now let's hear the transmitted signals at 3 different distances. So at 10 meters, sorry, you can hear a little bit some birds below. At 10 meters, 20 meters, sorry. And at 90 meters. As expected, you can see that for higher distances and higher frequencies, the recorded sound is dominated by the ambient sound. From these results, we can extract the attenuation law due to the habitat and simulate the listening distance of the recorder. So the listening distance corresponds to the maximum distance at which a sound is still detectable, meaning that is above the ambient noise or ambient sound level. We can see on this graph the listening distance of a recorder for a broadband sound at 80 dB that propagates in Jura at 11 AM in the morning in July. The horizontal axis is the listening distance or detection range, and the vertical axis is the frequency. The minimum listening distance is 5 meters for frequencies below 500 Hertz, while the maximum is at 150 for frequencies around 2 kilohertz. You can also observe that there is no unique listening distance corresponding to the biophony. The distance varies between 60 meters to 150 meters, depending on the frequency. So we just had a look on the listening distance for a single day and then single hour, 11 AM in July. The question now is, does the listening distance of the audio recorder varies over a year? And the answer is yes. You can see here the listening distance computed for every month of 1 year. And the whole circle, or it's not always a circle, is a frequency range for 0 to 1 kilohertz, 1 to 2 kilohertz, 4 to 5 kilohertz, and 7 to 8 kilohertz. And you can see that there is high seasonality variability. All patterns are different. There is high hourly variability. It's almost never a circle, like here or here. And there is a high frequency variability. Circles have different radius. So you don't hear the frequency at the same range, depending on its frequency. So now let's focus on 2 months, January here and so in winter, and June here in spring. And you can see that the listening distance is higher during the day in winter here than in spring. And this can be explained by the fact that during the spring, the ambient sound level is higher. Indeed, the rumor of the birds' calls increased the ambient sound level, which will reduce the listening distance of the recorder. And the listening distance is also much higher during the night, like here, than during the day when there is no anthropophony and very little biophony. This is during the night. And all in all, the radius Our listening distance can vary up to factor 3 in this case, which means that the area could vary by a factor of 9, which has a significant impact on the true density. So, to summarize, the listening distance varies depending on the ambient sound level and according the frequency and of the emitted sound. So low frequency and high frequency didn't have the same listening distance. We also observe a clear seasonal variation of the listening distance. So there is a clear need to take into account the variation of the listening distance to produce reliable acoustic richness and abundance estimators. And very interesting also is that this approach also works for other sensors, such as human ear or to estimate the effective detection radius, or for animals to study their active space. So now just a few words about a new tool. I would like to introduce the Scikit-MAD tools, which is a new toolbox for ecoacoustics that integrates the detection distance. With Juan Ulloa, we developed this package. It's a Python package. This package was designed to load and process digital audio, to segment and find regions of interest, compute temporal and spectral descriptors, and estimate sound pressure level. And now we can also compute the detection distance with this package. And here it's an example. So to estimate the sound pressure level, here on this graph, we can observe the average sound pressure level. Sorry, this is here. Pressure level the average sound pressure level per frequency band from 0 to 10 kilohertz or 11 kilohertz in this case. This was computed in June in Jura, and we can see that most of the energy, is concentrated in the lowest frequency band when anthropophony occurs. This is mainly due to the airplanes, but not only. And we can also observe the dawn chorus in the morning where we have more energy. Scikit-MAD allows also to simulate the sound attenuation and non-propagation distance. As you can see on this graph here, we simulate and observe the attenuation of the call of a spine-tailed bird at 10 meters until 20 meters, 200 meters, And at 200 meters, we can see that only the background noise remains, and you can see that the highest frequency is lost first before losing the lowest frequency. So this is an interesting tool to simulate propagation distance. Just a few words about something else, because this morning we talked about acoustic indices. Scikit-MAD offered the possibility to compute more than 60 acoustic indices. And here is an example of one-year variation of an acoustic index that reflect the acoustic abundance in French Guiana, which is our second long-term monitoring project. And on the horizontal axis, you have the hour from midnight to midnight here, and midnight here. On the vertical axis, you have the week numbers from 0, which is January to December. And then on the left, we can observe the acoustic abundance measured in the understory, while on the right, it's the acoustic abundance measured in the canopy. So the patterns are different, and we can clearly see the alternance of dry and wet seasons, and also frogs activity and insects activities. So thank you very much for your attention.
Thank you very much, Sylvain. That was fantastic. Again, and this is entirely my fault, we've massively overrun. So if you don't mind putting questions for Sylvain onto Slack, I know he's on there, so I'm sure he'll check and reply there. I'm very sorry about that, Sylvain, and thank you so much for that talk. It was brilliant. Okay, so very quickly moving on to the second part of this session, which should almost be finishing. So we've got parallel sessions again. The parallel session, which is going to be a talk from Paul Howden-Leech on how to use the wildlife acoustics software, and a video from Daniella Texeira on how to use Monitor, so that's template matching in the R software, and a talk from Stefan Kahl of BirdNet, which we've heard a bit of today about how to use BirdNet. That will be on in here. And then the main session, so for people online, the main session link, which is what you're on at the moment, we're going to physically move upstairs and we're going to have some discussions about problems and developments in species-specific monitoring. So I think there's going to be 5 minutes of audio-visual carnage while we do all that and move around. So quick loo break, but literally 5 minutes, and then back in here.
I think we're sorted to a point. So everything's a bit more stretched, but we're not going to bother worry about that bit. So I was talking about finding your call. That's where we got to. And this is the real problem, that you record 700 seconds worth of time, which is about 12.5 minutes, something like that. And you've got lots and lots of birds. How do you actually pull out those specific calls? There's several ways to do it. We can maybe just open up a file and we can kind of zoom in, and we just scroll left and right, go and find those particular calls. And we've got some chaffinches. That's a blackbird down there. There's a chiff-chaff, another chaffinch. But there's no way of quantifying this information. It's just in one long string. So we could write them down, maybe just go through a quick species list, but it's going to take us quite a while. So Kaleidoscope's got 2 elements. It's got a free element. Being from Yorkshire, I like that free elements. So that's what we're going to talk about first. And it all looks a little bit confusing, but it's got lots of tabs at the top. We can see this: batch, signal params, auto ID, cluster analysis, that sort of thing, acoustic indices. We can do lots and lots of things with Kaleidoscope, depending on what we want to extract from that information. So if we want to extract a call, we're interested in one particular species of bird, for instance, let's say a blackbird, then we can actually put in some parameters. We can get a blackbird call and we can say, well, what is the minimum and maximum frequency, and what is the minimum and maximum length of one of those blackbird calls? We can put some of that information in, and then what we can get Kaleidoscope to do is to pull out every single time something falls within one of those parameters. Now, what I've done here is just pull out everything. Let me give this a click. And what it produces is 2 things. One's a table and one's a sonogram. And wherever you are on the table, that's the sonogram that you're seeing. And what we can then do is maybe start labeling some of our birds. So here we've got 2 at once. We've got a chiff-chaff and a blackbird. So I've just labeled them straight to the table. There's a blackbird. That looks like a chaffinch to me. Yay, there we go. Another blackbird and something wren, maybe. Yeah, okay, another chaffinch, and so on. So this means that we're getting at a specific point a label. And because we've got lots of metadata that's stored in here, like the time, date, and location, we actually then get something that's actually quantifiable. The real problem with this is 2 hours on the dawn chorus, and there's 2,500 triggered events. So this kind of have a little bit of an issue if you're going to go through 2,500 calls every single day. But as a way of just extracting those specific calls, then this could be a kind of a useful tool. What we need to do is maybe jump into a few more measurements and parameters, perhaps. And some of these measurements and parameters you're probably very familiar with. I said I wouldn't mention bats, but a lot of these we tend to use for bats. So I'll quickly sort of go through them. These slides will be really quick. We've obviously got the call shape. We actually get on a sonogram a distinct shape. And when it gets to a point where you look at a call, so this is a chiff-chaff, we can see it's a chiff-chaff, because you can it's almost like reading music. We've got chiff-chaff, chiff-chaff, chaff, chiff-chaff, chiff-chaff. You can actually see it. This is a curlew at the bottom down here, and this is a blue tip. That sort of sound. Do lots of impressions, by the way. With that, we can change how we visualize the actual sonogram itself using changing things that are called the Fft window, which is a bit of a trade-off between frequency and time. So the higher the Fft window, the more detail that you get over frequency. The lower Fft window, the more detail you get over time. So here's kind of 3 quick examples, and I can talk to you about Fft windows forever if you really want me to, but I won't. Obviously, where the bird kind of sits on the frequency scale, that's again very, very useful. So we can fix these scales. We can either fix the frequency scale or the time scale, so we can actually start looking at specific measurements. So if we have a look at our 4 birds again, Hugh's disappeared, but I stole this cuckoo from his Xenocanto site. So this is this cuckoo down here at the bottom, but you can see where they all kind of fit in with that scale banding. But more detailed measurements, such as things like call duration. If we're looking at call duration, now we've got a long or a short call. So at the top, we've got a kingfisher. At the bottom, we've got a nightjar. So kingfisher's just doop sort of thing. This nightjar's for about 3 minutes. It's all right when you've got these extremes. But what happens if we want to actually do some scientific measurements? So are we looking at maybe an entire song? Are we looking at a specific phrase? Are we looking at a syllable within part of a song? You know, that could be some element of scientific research that I don't understand. But we want to be able to take those measurements. Other measurements that we might want to look at are the gaps. We call them inter-syllable gaps, so the gaps between repeated calls usually. So it's the gap from the end of a call to the beginning of a call. Generally in the bat world, and I like to put this over on the bird world, we tend to measure from the start of a call, include the call, and do it to the start of another call. And it's just because of how kind of sound travels. You know, when a call starts, buzzard, there we go. When a call starts, it's actually a very pronounced start, but it can actually fade out. So actually knowing where the end of a call is really difficult. But if you include the call as well, you still in effect get the same measurement. Great for things like alarm calls or woodpeckers, you know, something that's got a repeated phrase. Can't use it all the time, you know, we can't use it for every single bird call, but here's a couple of examples of a lesser spotted and a great spotted woodpecker. So a lesser spotted woodpecker, it properly drills into a tree. We've got a time between the start of one call and the start of another call of 43.42 milliseconds. Greater spotted woodpecker, a little bit slower, 65.13. So if we just got the pecks on a tree and we want to know which one, you know, is it lesser spotted or greater spotted, then we can actually use this measurement. The minimum and maximum frequency. Again, these are really good indicators of where the bird's actually going to be calling. Just so we know that we saw some sonograms earlier of harmonics. So these are harmonics. The actual call is this. These are naturally occurring acoustic harmonics. They're not necessarily the call. And the further away you are, or the quieter the bird, the fewer of these you'll see. The louder the bird, the more you'll see. So actually measuring this as a maximum frequency is actually really hard. You'd probably go for something down here at the bottom, like that. But we can do that in Kaleidoscope. What we can do is we can just draw a box, and a box gives you lots and lots of information. So we're getting a number in brackets. First number in brackets is the length of the box. Second number in brackets is the height of the box. And when I move the mouse, those numbers still stay down here at the bottom left-hand side. And we also get the start and end time here on this nice little graph that accompanies it. So from that, we can actually do specific measurements, and this is incredibly accurate. We can also copy those measurements and paste them into Excel if we really want to. I'll keep skipping over videos. Another kind of quite interesting indicator for manual identification is peak frequency. So peak frequency is the loudest part of a particular sonogram or a call. So here we've got, I think this is a buzzard again. So we've got this nice call, and then our peak frequency is at about 2.39 kilohertz. And it's applicable to any sort of call at the peak frequency. So we've got a nice, this is a wren here, and we can see where's the actual loudest part of that entire screen. Again, I think this is a gull of some sort, and we can see where the loudest part of that call is. And it tends to measure those frequencies to whatever is in that screen window. Obviously we can change that. So I've done this little demonstration here. I've drawn a tiny box to start off with. I apologize. There we go. So as I drag this across, you can see that graph on the right hand side is constantly changing to kind of highlighting whatever's on that screen. So it's constantly moving. And if I draw a little box around something, stop, draw a box around something, so that blackbird there, it gives me those specific measurements. And those kind of measurements are really useful in drawing out positive identifications and things. Which measurements should you use? Well, it depends on what you're actually measuring or what you're trying to identify. I tend to use a combination, and I tend to use the thing that makes a call stand out. You know, what's like the woodpeckers. Well, the first thing I would use is measure that gap between one peck and another peck. And if in doubt, you can always play the call. You know, if you're good at identifying birds by ear, then great. But the real kind of power within Kaleidoscope, we've got off the free stuff now, so if you're from Yorkshire, don't listen anymore. Is something called cluster analysis, which Kahl talked about a little bit earlier on, which is taking all your sounds, all your hours and hours of recording, terabytes of recording, and getting Kaleidoscope to group them together into sounds that sound the same. So this is sort of machine learning, but it's not going to get any better, if you see what I mean. So I've got a quick video here of me having processed a batch of data. So I think this is just a small batch of data. I've only got a couple of thousand files, maybe about 1,700 files. And you can see here, I've got these cluster groups. That one goes up to 19. So I've actually got 20 because it starts at 0. So this is a really, really small data set. But I'm going through this cluster 0, and you can see it's picking up this particular call here. So I don't know what that call is. I could go and play it. But basically, if I want to label this really, really quickly, I can bulk label this entire group. Oh, I've looked at it in context. That's what I've done here. That's me not looking at the screen. So you can see here, I'm looking at it in context. You know, where is it within the file? I've only got a tiny bit of a call. And then I can label it. So I can bulk ID. I've called it something really imaginative. Look at that, bird 1. And it's identified all those very, very quickly. Now, they're not all right. Some of them will be wrong. You know, I could put my mortgage on the fact that some of those will be wrong, but the majority of them are correct. So it's identifying roughly where your sounds are kind of living. You can see here that the next cluster group and the next cluster group has kind of got the same sort of signal, and that's just because of how the clustering works. Cluster 0 is the group with the largest number of vocalizations that sound the same. Cluster 1 is the group that sounds as like cluster 0, cluster 2 like cluster 1, and so on. Eventually you move away from that cluster group. And you also will have multiple groups of the same bird or the same call, because there's things like this background noise which changes. Your bird could be calling with another bird at the same time. So you probably have a group of more than one species at the same time, which is put together, because it doesn't know the difference between a blue tit and a wren calling at the same time. It knows that it's got those two signals. It's just looking at, in effect, pretty pictures on a screen. And finally, how you can kind of move on from that, if you get really into this, you can build your own classifiers. So you can build classifiers to pull specific birds out of much bigger data sets. The advantage of that, or shall I say the disadvantage of clustering, is if you've got a bird that calls once, it's got nothing to group it with. So it'll go, well, it's obviously just a rogue noise, but it could be that one rare bird that you're really after. So the way to compensate for that, or to get around that, is if you build a classifier yourself and run that against that same data set and that bird called once, provided it's a nice call, it'll probably pull that out. Okay, so that's kind of what it is. And this is my example here. Instead of it saying clustering, it's put in what species it is. And this is a wren. So it's got that one right. The output, well, the output's a CSV file, and the output tends to have the name of your file, the time and the date in a 24-hour and 12-hour clock. These lines go on forever and ever and ever, and then you get a manual ID. So it basically means you can do your Excel or R wonderment, whatever you kind of fancy. I'm just about done, so I just want to point to a few resources we've spoken about today. Xenocanto, brilliant resource. Go onto the Xenocanto website. It's got every call of every bird in the world you can imagine, from every part of the world. It's an amazing resource, and it's a wonderful community. If you record bird sounds, put them on there. People will help you out, help you identify them and things like that. So go there. UK Bird Sonogram ID. Watch this space. Myself and Carlos might be working on something. It's supposed to be out this year, but Carlos isn't here, so it might be next year. Anyway, and the Wildlife Sound Recording Society, which we also mentioned earlier, they're a wonderful place to go to. Subscribe to it. If you haven't got any equipment and you want to kind of borrow some, they'll loan equipment out to you, sound recording equipment. The World Wildlife Fund as well. I've done a whole raft. We've got a whole raft of information on bioacoustic recording. Again, we mentioned that earlier. And then I know I've highlighted it in blue, but we've done lots and lots of webinars on every aspect of sound recording. Our last webinar was on social calls of bats, and we had 1,000 people turn up from all over the world. It was amazing, really good fun, really relaxed. And all the previous webinars are on there, including our first one on acoustic indices. So please go on there, and I offer loads of free training as well. So please go on there, and I think that's it. Oh, thank you.
Very good. So thank you so much to Paul. Oh, we've got a nice.
That's cool.
Next up, maybe we'll move on to the second presentation, which is Stefan from Cornell looking at the BirdNet, and then we'll have a discussion. As we're running so late, what I'm proposing is, hang on a sec, let me just sort out. Stefan, thank you. I'm just switching screens. You've currently got an Indian deity instead of you on the main screen. I think, yeah, if you're both happy with that, we'll have the presentations back to back, and then we've got some time for discussion. And then, as we're running so late, if people are happy, I'm proposing we'll share Daniella's pre-recorded presentation. We'll put it on in a coffee break or something at another time, because I think people probably need to eat, feed their children, that kind of thing. So bear with me one moment. So, Stefan, over to you.
Hi, thanks. I am just trying to share my screen. Can you see the slides?
We can't hear you at the moment. Do you want to?
Can you hear me?
So I'm outputting to.
Yes, we can hear you. Yes.
Oh, okay. I'm unmuted, so okay. So there's at least someone who can hear me. That's good.
So simple. Here we are. Here we are. Let's try this. Oh.
If you mute your mic.
Yeah. I think Teams could hear Stefan, but we couldn't. Let's try now.
Speaker volume low.
Or is it because that microphone's plugged in?
No.
Oops.
I'll just unplug this mic for now. If I mute this mic, then you can turn that on and we might be all right.
Yeah. Okay. Try now.
Okay, can you hear me?
No. Oh, yes.
Is it working?
Something there. Stefan, can you say something again?
Yeah, I would like to share my screen if the audio is working. Can you
Really
hear
bad
me?
connection.
Yeah, that is not a good connection. Something else. Right. Mine's off, yours on, that's on. He's unmuted. Microphone's in, output is right.
Yeah. This is on HDMI, the output.
Oh, no. No. Let's see why that's not working.
Is it because we're on the free Wi-Fi as well? You're on the free Wi-Fi on the proper one?
Yeah, you can't go on the LAN here.
That shouldn't really impact it though.
Try now. Could you try again, Stefan? Just give us something.
Sure, check. Can you hear me?
Yeah, we can hear you, and it's a terrible connection.
Yeah.
Okay, so is it me or not? Because I can hear you fine.
Stefan, can you just check your microphone settings?
Yeah, sure.
My batteries are better on there. This is great.
MMU did warn us that
But
they
that
did not
looks
support
fine.
hybrid conferencing.
Via Teams.
No, that looks fine. It's using...
Can the people on Zoom, on Teams hear Stefan okay? Because for us in the room, it's really, really breaking up.
We can hear Stefan, yeah, so it's just us.
Yeah, put the chat up. There's something in the chat. I can see there's a little bubble.
Yeah, no, we've got the chat monitoring on another thing.
Yeah, they can hear it. They can hear him very well. It's just our room.
So I think it's the EduRoam connectivity because they won't allow these laptops to go on.
Yeah.
Keep trying.
Say something else, Stefan.
Something else.
Yeah, it's the connection.
It's such bad connectivity. So the only thing we can do, this is insane, but we could switch back to the Power PC.
You only need to log in onto that. So
Yeah, all you need to do is switch back to that and just unlog from me. That should, and at least then you've got a wired connection. The dance of connectivity. Yeah, so now you should switch back to that. Now I'm no longer.
So
that should be that sharing now. Yeah, that's right.
Yeah.
So now that PC is in control and that's wired into the network. So that should. So if you just click back on Teams.
Jesus. Okay. So that's that.
You click on Teams and then that should just be there. We should be good.
That's the parallel workshop.
That's what was the confusion, right?
Yeah.
You've logged in twice, one in each room. You each logged in the different rooms.
Yeah. So it's that one. That one.
Yes. Yes.
Hey.
Say something, Stefan. Talk to us.
Check. Is it working? Can you hear me?
you need to switch back to you unplug the HDMI from your.
No, because we've switched on here.
Oh, yeah.
Why doesn't that work? Wait.
Then try the output. It's muted.
Try now, Stefan.
Say something. Check.
Have a go. Because Teams has been muted. No, it's not got any microphone plugged into the PC.
We don't need a microphone. We just need to hear Stefan.
It's the speaker output.
Yeah. So
if you go click on more, device settings, audio output, speaker. What's that one? It's only got one option, and it's muted.
There we go.
There we go. Let me just mute all of these.
Mute yours.
I think it's.
Right, that was weird. So check again. Can you hear me? Because I can't hear you.
Yes.
All right. I'm trying to share my screen.
So thank you so much, everyone, for bearing with us. It's a great pleasure to have Stefan Kahl with us from Cornell. Stefan, are you able to share your slides? Yes, fantastic.
Yes. Can you see me?
And we'll now pass back to you,
slides
Stefan.
and hear me? All right, if you can, that's good. Yeah, thanks for having me. Thanks for letting me introduce the BirdNet Analyzer for scientific audio data processing. And I'll make this a rather quick presentation, but I just want to briefly introduce what BirdNet actually is. And yeah, generally, what do you think if you hear BirdNet? It's like, yes, it's an algorithm. It's a detection algorithm for birds. It's a neural network, hence BirdNet. But it's actually more than just an algorithm. And I think most people will be familiar with the BirdNet app or the BirdNet online tool that we have. And so like typically when we talk about BirdNet, people think about the app. But BirdNet can also be an embedded system or running on embedded system, monitoring in real time. And BirdNet can actually also be a research tool. And this is what I'm going to focus on. This presentation is like BirdNet, a research tool, and how we're going to make it accessible to a broad audience. But how does it actually work? And I think this is pretty much straightforward for anyone attending the workshop. I think you've heard that like a couple of times. You need raw audio data, and then you're going to split this up into 3-second chunks into spectrograms, and then we're going to feed these spectrograms into the BirdNet algorithm, which is a deep neural net, and then this deep neural net will output species probabilities. And in this case, yeah, let's say it just predicted 3 different species for this call. And so what we can now do is take these predictions and pass it through a eBird occurrence mask and say, hey, based on the metadata that we recorded, based on location, based on time of the year, which species is actually probable. And if we assume that this recording was made in Ithaca in New York, we say, yeah, well, it's very likely that it's a cardinal because the indigo bunting might not be there, and the goldfinch is a European species, so there's no chance to encounter those in Ithaca. And so that's like really rough overview of how things actually work. And I also want to introduce like the vision that we have for BirdNet. And again, this is more like the research part of BirdNet, not necessarily the app. And so for the next few years, what we really want to focus on is like transforming BirdNet as it is right now into BirdNet Plus, and open sourcing and putting that out there and making it accessible for a broader audience. And what we want to do is like really improve. Of course, we always want to improve the models that are that we provide. We want to go up to 5,000 bird species. We want to add amphibians and mammals to it. We want to have a smaller computational footprint because we want to go on embedded devices, and we also want to go beyond classification, more towards representation learning to do similarity search. We want to support a bunch of devices actually. So like right now, the most common one, like the one that most people are familiar with, is the app. But we also want to support microcontrollers like Arduino boards, Raspberry Pis, of course mobile phones, but also workstation PCs and even cloud servers. So like the entire scale from tiny to extremely big, we want to cover all of them. We want to release a bunch of interfaces What we really want to do, well, it's kind of like what I'm envisioning is BirdNet as a service. You can say, hey, I need the analysis, I need an analysis tool, and there's a service for it, and you can just access it, run your data through it, and get some results. There's hopefully at some point going to be an R interface. We want to interface, of course, with Raven Pro or Raven X, which is like in-house analysis software. We want to provide a bunch of web tools that lets you analyze actually your results you got, and we want to natively support all kinds of operating systems without the need for any setup. And then the most important part, and the one that I'm going to focus on in this presentation, is like open sourcing BirdNet, publishing our code on GitHub, actively managing an open source community that hopefully evolves around this published code, and then let the community work on other cool projects. And in this presentation, I'm going to introduce our most recent repository on GitHub. And so what we did is we published our models and the inference code on GitHub, and it's open source. It has a non-commercial license, so everyone can use it. Not necessarily for a commercial project, but like for research, education, you name it, you can use it. Just go on GitHub and then search for BirdNet-analyzer, and then you will hopefully find the web page that I have the screenshot on the right-hand side. And so what you need to do to actually run it, and so this is kind of like a bit of like a really small, really brief tutorial on how to do things. Typically, what you have to do is you have to clone the repository to your machine. And then if you did that, you can run the analyzer by just providing an input path containing all the audios and then an output path where the results should go. And so this is like a really simple interface. This is a command line interface. This is still really simple. And it provides a few other options which make it a bit more flexible than that. You can actually provide latitude and longitude and the week of the year to restrict the species list that you're looking at, because not all of the 2,400 species that we currently support occur in every location. So like it makes sense to restrict the species list based on latitude and longitude. You can provide your own species list as a parameter. You can adjust the sensitivity for each detection, set confidence threshold. You can specify certain output formats. We're supporting CSVs or simple text files or Audacity or Raven selection tables. You can actually specify the number of CPU threads you're going to use. You can actually do distributed computing, which would mean that if you have a machine that has like 16 cores, you can actually run 16 files in parallel, which should speed up things. And then you could even set translated species common names if you wanted to. And so this is a command line interface. And so typically as a computer scientist, and I'm a computer scientist, you would say, yeah, that's super easy. That's easy to use. But it's actually not for many people. And so we decided to build a GUI, graphical user interface, which does the same thing. It's not super pretty. It's very basic, and we're still trying to improve that. But it does the same things. You can set like input and output paths and like configure all of these parameters. And the interesting thing about this is you can actually download an executable that runs on Windows, which does not require a setup. So you can actually download a BirdNet version that does not require a setup, that has GUI, and then you can start and analyze your data collection. We're working on like this repository, like we're working on all kinds of different interfaces, and I mentioned that before. We want to support different devices and different use cases, make it more accessible. And right now, what you can do with this repository, you can actually also run it in a Docker environment, which means you can actually deploy it to a cloud environment and let it run in Docker, let it run in a distributed way. You can also host a server, like this repository contains a server implementation that lets you host your own API if you're interested in setting up a web service that you can query. And then we're already working on an R interface, which will soon release. And we're also working on a BirdNet version in JavaScript, which would actually let you run BirdNet in a browser. And so we will continue to develop these additional interfaces to make BirdNet more accessible, to let more people use the tools that we're building. Some people actually made use of the repository, and that's what I want to introduce in the second part of this presentation. It's like some community projects that evolved around this repository, because I mentioned before, you want to like actively manage an open source community and try to develop BirdNet with this community and trying to support these really cool projects. And there's 4 of them that I'm going to introduce briefly. First one is HiQbox. Second one is BirdNet Pi. Another one is BirdWeather, and then the last one is the EcoPi Bird. And so if you go on HiQbox.com, you can see that this is a device that you can put in your backyard. You can buy it online. It's been released. I think it's $249, and it has a Raspberry Pi compute module in it that runs BirdNet in real time. It needs a Wi-Fi connection, and it needs a power supply, so it's not totally independent. But what it does is it will record 24/7. It will detect 24/7. And then there's a web page where you can access the detections, and it will show you like the species, like you can see here on the right-hand side. It will show you the species that are detected in your backyard, which is pretty neat, I think. The same thing, but not as a proprietary device, but something that is open source and that is more like the do-it-yourself version of HiQbox is BirdNet Pi, which is actually like really cool project. Like if you have a Raspberry Pi and a microphone lying around, make sure to go to birdnetpi.com, run this command that you can see on the top, flash it onto an SD card, and then have your 24/7 monitoring station in your backyard on your Raspberry Pi in real time. And it also comes with a web page that lets you see statistics on the species that were detected, lets you listen to the live stream of your Raspberry Pi, and lets you do all kinds of cool stuff. So it's like really a project that has really come a long way, that has a lot of really cool features. It's definitely worth checking it out. Like if you have a weekend and you want to kind of like tinker around with a Raspberry Pi, make sure to check out BirdNet Pi. If you do, then also make sure to hook up your BirdNet Pi to BirdWeather, which is another community project which analyzes live streams from around the world. And on this map, if you go to birdweather.com, on this map, you can see the red icons. Those are YouTube streams. The blue ones are audio live streams that are being analyzed using the newest BirdNet models. And the green ones are actually BirdNet Pi. So those are actually people running a Raspberry Pi with BirdNet on it and submitting results to this page. And you can listen to some of these stations. You can click through the detections, give a thumbs up or thumbs down if it's a true positive or false positive, and kind of like see which species occur at which location over time. And again, this is real-time 24/7 monitoring at your fingertips. And then the last one is a project that we're currently working on with a German startup Öko4, and developing a completely independent recording device, which also is based on a Raspberry Pi, which is solar powered, has a microphone on it, and submits results using a small SIM card, and can run 24/7 detection. But since it's solar powered, it makes sense to have a recording schedule, let's say 10 minutes every hour or something like that. And it runs BirdNet on device. You can provide your own species list, and then you can submit results. And these results that these stations will submit can be visualized in an R-Shiny app that Öko4 built, which is super interesting. And so hopefully we'll be able to release that relatively soon. And so then on this page, you can also go through detection, see time plots, you can verify detections, see locations where certain species are detected. And so with this recording device that I just showed you, we actually do have 2 kind of smaller success stories. And in collaboration with the Vogelwarte in Seppach in Switzerland and the German startup Öko4, we monitored like different locations and we're looking for species that were thought to be extinct, the rock ptarmigan and the little owl. And we actually managed with those devices, with the help of BirdNet, to detect these 2 species in these areas, which is super cool and kind of like showing that they're not extinct in these areas, and all doing that with on-device monitoring without the need to do recording first and then processing. You can do this in real time on-device, which is super cool. And then, so this is my last slides, like how can you actually contribute if you wanted to, since it's a GitHub repository. what you can do is submit a feature request by opening an issue, or if you want to contribute code, you can actually do that and submit a pull request. Again, we will try to actively manage the open source community. We're actively managing the repository. And so if you have a feature request, if you have a question or anything, just let us know, and we'll make sure to implement that and release it for everyone. And with that, thanks again for having me. If you want to know more about the project, just make sure to go to birdnet.cornell.edu, or if you have a question right now, I'm open for it and I'm happy to, talk. Thanks.
Thank you, Stefan.
Thank you so much, Stefan. Made absolutely extraordinary advances in the last few years. How are people feeling? It's now 5:27. Should we have at least like 10 minutes kind of question discussion? I'm sure people are here presumably because they want to learn how to use this, and you may have some questions. Paul, would you like to come? Actually, we haven't got the camera anymore, but if you come back up here for the microphone.
I'm probably better off camera.
Okay. And if there are any questions from Teams viewers, please put them in the chat and we'll field them here. But yeah, any immediate questions from the room?
This is more of a following.
Do you want to? Sorry, can you use this so we can?
Oh, sorry.
No. Are we working? Do we still have a microphone?
Yay.
Thank you. Thanks, both of you, for interesting talks. It's a question about Kaleidoscope Pro, actually. I was just wondering if it would be able to cluster, the cluster analysis, would it be able to cluster different populations of the same species based on similarity, or is it not that sensitive?
It depends what it is. So, might be that microphone. I'll go grab it. I'll go grab it. Why not? Yes, it'll depend on what that call is. So basically, if you've got a bird that makes more than one call type, then yeah, it will quite happily put the different call types into. So, for instance, starlings make about 30,000 different call types, and they're in lots and lots of different groups because they all do different things. If you're looking at something that's going cheep, 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 and then something that's going the same bird's going cheep, 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 it might throw it together because it is a little bit it's very, very close. There are ways of actually doing an advanced classifier to try and split those two up. So there are ways of doing it, but I can't promise that it will definitely do that. And the next thing a lot of people ask is, can it do individual birds? I'm like, yes. I'm not saying it can. It probably can't. So.
Thank you.
Any other questions?
Any questions from anybody online?
Maybe people are saturated.
Yeah.
Okay. Well, I apologise again for the relentless technical issues. It's all part of the learning. Huge thank you to Stefan for joining us.
Yes, thank you, Stefan.
And thank you to Paul for his multiple appearances. So you've got all the software is available online, so you can go and explore to your heart's content. If you've got any other questions about this session, particularly for these speakers, please put them in the chat on the Slack channel for this session. And of course, we'll make some kind of group afterwards to keep discussion open. Okay, so have some air. Thanks again, Stefan.
Thank you, Stefan. Okay.
Well done for your multiple juggle.
Hi everyone, my name is Daniella Texeira. I'm a research fellow in applied ecology at the Queensland University of Technology in Brisbane in Australia, and also an ecologist with the non-profit organisation Bush Heritage Australia. Thank you for coming along to this talk, and I'm really sorry that I'm not there live today. The time difference is a little bit tricky, but please feel free to email me if you have any questions. I'm very happy to chat. So today I'll talk to you about some of my work using the monitor package for bird call recognition. Before I begin, I'd like to acknowledge the Turrbal and Yagara people as the traditional owners of the lands where QUT, my university, now stands, and I pay my respect to elders past, present, and emerging. So what is Monitor? So Monitor is a package for animal call recognition, and it's implemented in R or RStudio. So R is a programming language that's fairly commonly used nowadays amongst ecologists, and it is relatively accessible to people like me who come from ecological backgrounds, not computer science backgrounds. And yeah, it's fairly widespread, and we use it for a lot of data analysis these days. It's open source, it's free, and once you get your head around it, it's not too difficult to use. So Monitor is a fairly simple form of recognition. So it's not any kind of complicated deep learning method. It's basically just template matching, but it does provide two options. So it provides spectrogram cross-correlation, which some of you may have come across in the literature. And it also provides binary point matching, which is actually what I'm going to talk about today, just because in my work, it seems to perform a little bit better than spectrogram cross-correlation. And essentially that's what I've been using, but very similar, and the functions in the package are almost exactly the same for the two methods. So the binary point matching method that is in Monitor is a modification of that was developed by Michael Towsey and others a few years ago, which is referenced in that bottom paper there. So why would you use Monitor? There's so many options out there these days, and which one do you choose? It can be really overwhelming and unclear as to which method you should really use for sound processing. This is a very general, simplified take on things. So please, don't hold me accountable to all of this. I know there's all of this can be debated, and there's certainly methods here that are missing. This is just a very quick thing that I cobbled together. And I just want to say I actually do use all of these options. So I do use commercial software for certain projects. I use custom, very advanced sort of machine learning for other options where I work with computer scientists. And then I use Monitor for other projects. They all have their pros and their cons, and I think your choice about which one to use really needs to come down to things like your project objectives, who you'll be working with and what their skill sets are, what your budget is, etc., etc. So Monitor, I think, is fairly user-friendly once the code is written, and fairly accessible to ecologists if they're able to use R, which many people are these days. And a real bonus is that it's free, so you don't have to blow your budgets on buying any fancy software. Just some terminology. So you'll hear me use these words template and survey quite a lot. So the template is basically just the reference call, so the isolated reference call that you feed into Monitor, and that's what it will use to process the sound files. And those sound files are called surveys. So just to clarify that, because you'll hear me say that quite a bit in this presentation. And this is basically what Monitor does. So here we can see a spectrogram of a nestling call of the glossy black cockatoo, which is one of the species that I work on and have the great fortune of being quite involved in their conservation. So Monitor basically takes the spectrograms, the template, and creates this binary point template there that you can see on the right. So essentially it just creates on and off points. So the blue is off and the orange is on, hence the word binary. So it's fairly simple, but actually choosing which calls to become templates is important. And there's a lot that kind of goes into the parameters that sit behind the templates themselves. So once you have templates created, Monitor will basically step those along the survey files. And then for each time bin, it will score the similarity between the templates and the surveys. So the higher the score, the more similar the template is to the survey. If you can see that figure there on the right, I'm really sorry, those axes labels are tiny. So if you can't read them, so the x-axis is just time. The top figure is a spectrogram, so frequency on the y-axis, and then the bottom figure has score on the y-axis. So the score being the similarity between the template and the survey. The three colors that you can see, the red, blue, and green, those are three templates that I happened to test on this survey file, just for this example. If you can see on the bottom figure there, there's a green horizontal line. So that's the score cutoff. So that's something that you as the user actually set. And anything above that line, Monitor will return to you as detections. Anything below that line, you can see those peaks and troughs. Monitor is detecting that there's sound events there, but they're not above the score at which it would return it as a detection. So getting that score cutoff right is actually really important for this method, because it trades off your false positives and your true positives and your false negatives and all of that. So the higher you have it, the fewer false positives you have, so you should get better precision, but then you're probably going to start missing some calls. If you bump it right down, you'll have better sensitivity, so better recall, but you're likely to get more false positives. And deciding what that where that should sit for your particular project is based on so many things that I don't really have time to kind of talk through in detail today. But for this example, for glossy black cockatoos, I didn't need all of the calls on a day, I just needed a few. So having better precision and a higher score cutoff was important. You can see there the boxes have detected 3 calls of the glossy black cockatoo, so those true positives. They're the peaks above the score cutoff line. But it's also missed a few calls, so those are false negatives. Those calls are given further away from the microphone, so they're much lower in amplitude, which is okay for this project. But again, if you needed to detect calls that were far away from microphones, you would really need to lower that score cutoff to hopefully detect those as well. Like I mentioned earlier, actually figuring out which calls should become the templates is quite important, and I don't really have time to talk through all of the ins and outs of creating templates, but I just want to say in my work I did a really comprehensive pilot study based on all the different call types that these birds give at nests at 3 different amplitude cutoffs, which is sort of what determines the on and off points in the template. There's a lot of parameters that you can modify. And really the takeaway from all of that was that performance is really quite variable, surprisingly variable between templates and between different score cutoffs as well. So I spent a lot of time thinking about this and testing a whole range of parameters. And ultimately, the templates that performed the best were not actually the ones that I would have thought were the best ones initially. So I think part of the temptation with a method like Monitor is that it's pretty easy to just feed in a template or two and sort of plug and play, so to speak. But it's important that you recognize that those might not be the best templates, and often what we think are the best templates aren't. So if you do have the time and the capacity, I really recommend testing a lot and then verifying those against some, basically a test data set that you know that's fully annotated, or at least you know where the birds are present in those sound files, so you can actually measure performance and understand what you might be missing. Again, there's a lot in that. I don't have time to talk through it all today, but just, yeah, think about your templates carefully, I guess is my word of warning. So for the glossy black cockatoo, these were the 2 templates that basically were the best performers. I just selected 2 to kind of keep it reasonably small. The more templates you have, the longer it takes to run. So again, it's a trade-off between speed and performance. These 2 performed pretty well. They're both nestling calls. And this is the final output. So I brought the sound file into Raven, and the blue boxes are what Monitor has detected. So I'll show you how I get to this point in a second when I do a little demo. And yeah, all the details of this is in this paper that we've just published. So big shout out to my co-authors, Simon, Richard, Martine, and Berend. This is a lot of work by all of us, and yeah, thank you to those people, wonderful people. If you have any questions about what's published in here, again, just please feel free to reach out and get in touch. And all the code that I'll show you now is available on my GitHub. So you're free to download it, use it however you want. If you do use it for a project, please cite the paper. And yeah, I'll give you a quick demo now. Hopefully this works. Okay, so if I just show you what I've got here, so I've created just a little demo folder. So there's basically two different folders here I've called surveys and templates. So the templates are those two templates that I just showed you in the presentation. So they're the two ones that after all of the pilot testing performed the best. And I actually created what I've called a stack here. So those templates, I don't need to read in as individual like WAV files anymore. I just kind of read in this stack, and it has all of the data associated with those templates, like what their score cutoff should be and all of that. I've just created a survey folder here with a single 5-minute sound clip just for the demo. But if you had lots of other files to process, you could obviously put them all in a folder like that. All right, so if I move over to R, so this is what the recognizer essentially looks like, or the recognizer code. I don't have time to kind of go through how to use RStudio, so I'm just going to kind of press forward with the assumption that this all kind of makes sense to you. I'm sorry, hopefully I've commented it enough and it's clear enough. So I'll just set my working directory and I'll load the few packages that we need. So monitor being the most important one. So with the templates, so I'm just going to show you how you would create the templates if you didn't have them in that stack that I just showed you. So if you were just kind of bringing them in an initial project, and you just wanted to kind of have a look at them and modify the parameters and see how they impacted it, this is what you would do. So I'll just uncomment those lines so that they're active. So this is reading in that first template WAV file. If I do that, there we go, and you can see that there it's created the template there, and likewise if I do that for the other one, same thing. You would then have to combine them together with this combine bin templates function. So I'll just run that. You can see the table down here where it's combined them. And if you wanted to modify the score cutoff, you can use this template cutoff function. So I'll just do that, and you'll see, if I go down here, that the score cutoff was 12. That's sort of its default, but I changed it to 18. But because I've done all of that pilot testing and I know exactly what I want these templates to look like, I'm actually going to comment that out. So it's inactive, and I'm going to reload this with the stack that I showed you before. So I'll bring that in. There you go. And these are the same templates, but you can see that I've got these score cutoffs that are specific to the template. So 15.8 and 19.2 for the other template. So that's the score cutoff at which those templates performed best. Okay, so down here, I just basically point to the directory where those survey files are. So in this case, it's just that one 5-minute sound file, and it should, yep, it'll read it right there, which is great. And then to actually run the recognizer, it's just this single loop here. So the actual recognition part is down here, these lines sort of from 72. This function bin match, so basically binary match, is where all the magic happens. And the rest of this code is really just finding those peaks. So if you remember to that earlier figure where there were those peaks and troughs, so basically it's this function find peaks is finding those peaks, and then get detections is looking for the ones that are above that score cutoff. And yeah, basically then just put that all together into a data frame and export it as a CSV file. Just one other thing I wanted to highlight here. I have this section here, interval equals 60, which is actually really important. So basically what I'm doing is reading the sound files in as 60-second snippets. So my computer memory, I'm just running this on my laptop at home, doesn't completely get overwhelmed by bringing in these, multi-hour long sound files. So that's just something that I found has sped this up. Okay, so I will just run this, and it shouldn't take very long for a 5-minute file. So you can see down here, it's sort of going through it. Great, so that's done. So now if I go over to the folder where I had everything stored, so I'll go to the surveys, you can see there that it's created the CSV output file. So this is what it looks like. It's basically got the template that the detection came from. So let's open that. It's got the score, so the similarity score, and the time at which that detection happens. So here we've got, 26 detections. So at the moment, this is great. This is, we can use this, but what this will be doing is different templates. So in this case, the 2 templates that I've used, if they both match the survey above the score cutoff, both of those will be returned as detections. So what we actually need to do is remove those duplicates so that we only keep, say, the highest scoring detections. There is a function in Monitor called, I think it's called best detect, or time align, I think it is, which I haven't used just because for various reasons, I've actually just written my own script to deal with that, but just so you're aware that does exist. So I'll just close that. And I'll just go back to R and I'll just show you this script that I've created here specifically to read those CSV files in and output them as text files that Raven Pro can read. So Raven Pro is sort of the software of choice for me. I think it's great, and the verification process is pretty quick in Raven Pro, particularly 1.6. The new features are really great. So you can, if you want to, stay in R and verify everything using Monitor. You can go through all of your detections and do your yes, no, like what is actually the true positive or false positive. But I prefer to bring everything into Raven. So I'll just run this script, which will give me outputs in the form that Raven needs. So, oops, go back. So I'll just run all of this. I'll read in the output files, which in this case is just a single CSV file, but this would loop through all of them if you had them all in a single file. And then this loop here, basically just cleans that CSV file. It identifies duplicate detections, removes those duplicate detections, and then adds some columns with exactly the right column headings that Raven Pro needs. So it has to be specifically this. So I'll just run that. Okay, and if I flick back here, we'll see that there's now a text file, and it should have fewer rows than what that CSV has because it's removed those detections. Okay, so if I just quickly go to Raven Pro just to show you what this looks like, I'll bring that survey file in to Raven Pro, and if I open the text file as a selection table, there we go. So that's the 5-minute clip. The blue boxes are the detections by Monitor. And yeah, it's pretty good. You can see it's detecting all of the loudest calls. These fainter ones, most of those actually are the calls of the glossy black cockatoo as well. So it's missing a few, but that's okay for this particular project. We really just needed to know whether these birds were even there on a given day. So that was fine. So that's basically it. It's pretty straightforward once you've kind of got a basic understanding of how to use R Studio, and the code is all there. Hopefully I know that was a really quick overview, but hopefully it gives you an idea of what Monitor can do. And yeah, like I said, I'm really happy to chat and take questions via email or otherwise. And I'm really keen to see what you guys might come up with using this package. Thank you so much. And yeah, hopefully talk to some of you soon. Bye.